Hello everyone. In today's class, we'll talk about the structure of flagellum as well as the classification of bacteria on the basis of arrangement of flagella. First, let's talk about the general characters or the general features of flagella or flagellum. The word flagellum is used for the singular, and for the plural form, the word flagella is used. Now, before going to the structure, let us see the general features of flagella. A flagella, you can see here, it's a filamentous structure or a hair-like or lash-like structure that is present on the bacterial cell wall. Like this in the previous class, which I have mentioned, that is present on the bacterial cell wall and which is playing an important role for the different physiological functions. So how it was taken? It was taken from the Latin word. It is actually a Latin word. Flagellum is a Latin word which means whip. The term flagellum is the Latin word which means whip, and it indicates the long slender structure of flagellum that looks like a whip or that resembles like a whip. And these flagella are basically or primarily essential as an organelle of locomotion. Besides, also they help in gathering the food and circulation as well. Okay, these flagella that are occurring in different organisms have different structures. That means based on the species or based on the organisms, the flagella are different in structure, different in mechanism, different in movement, as well as they are different in their functions. We are talking about the structure of flagellum. The structure is different in case of prokaryotes as well as in case of eukaryotes. Like even within the prokaryotes also, the bacterium flagellum is different from the archaeal flagellum. Now similarly, when we are talking about the composition and mechanism of the flagella, the formation are also different. So basic structure we'll discuss here. Now the basic structure of the flagella is consisting of filament, hook and the basal body. The filament is a thin, long hair-like structure which is present in mostly in case of all 98% of all the structures of all the flagellum. Right. So it is attached to the hook part of the flagella, and this movement of the filament is controlled by the motor, which we'll study now. Movement is So the filament movement is controlled by the motor body or the basal body which is present in the inside the cytoplasm. That means this filament is observed outside the cytoplasm. The next part is the hook, which is a connecting part between the filament and the basal body. Hook is otherwise called as anchoring structures. Anchoring structures. Now these are very short or curved or we can say tubular structures that will connect the basal body to the filament, long filament, and the most important function of this hook is that it move in different orientations for the different functions. And major function for the hook is like in the assembly of flagella or flagellum, in the assembly of flagellum. This hook is composed of numerous protein subunits now which is forming the polymorphic supercoil structure. Because of the protein subunits that are present in the hook, these are forming the supercoil structures, and this structure is present near the cell membrane. It is not inside the cell membrane, but it is present near the cell membrane in all the cells. But the shape and exact composition, like it can be of like this exact hook shape or like curved shape. So it can be of different shape, and also the composition may slightly differ between the different cells. When we are talking about the compositions, filament also is a macromolecular structure which is composed of hook proteins and flagellins. These hook proteins help in the attachment to the of filament to the hook. Last one is the Bessel body, or we can also call it as the Motard device. Okay, this is the only structure that is present inside the cell membrane or within the cell membrane. Out of the three structures, filament, hook and Bessel body, this is the only structure which is present inside or within the cell membrane. What are the main functions of this? Actually, the Bessel body is a rod shaped structure, like with having the rings here. You can see the rings here. These rings are also named differently. These are the rings, and the main component of the Bessel body will differ in different cells. The rods which are present here, these are the rods which are present here, are responsible or act as reversible motor which help in propelling the filament in different orientations and also for the different functions. And this Bessel body is also essential for the transfer of flagellar proteins because it is present inside the cell membrane. So how it is transferring? It will transfer the flagellar proteins from inside the cytoplasm, from the cytoplasm to the hook part of the filament or the flagella during the assembly. Which is forming the assembly here, hook will help out in the assembly of flagellum. So the proteins whichever are present in the cytoplasm that are transferred with the help of Bessel body to the hook part. This was the basic structure of flagella, and based on the arrangement of flagella, the bacteria are classified into different groups. Now let us move on to the classification of bacteria based on the arrangement of flagella. Based on the arrangement of flagella, the bacteria are classified into different groups like Atrichus, Monotrichus, Amphitrichus, Lophotrichus, Cephalotrichus, and Peritrichus. The first one is Atrichus, the bacteria which does not contain any flagella. There are no flagella at all at the cell membrane, and they are called as A. A means no. Atrichus bacteria. The example for this is Shigella. So we are talking about the Monotrichus. Mono means single. That means only one. The bacteria which is having only one flagellum at the polar end are called as Monotrichus bacteria. Example, Vibrio cholerae. Now Amphitrichus, the bacteria in the diagram itself you can see if the flagellum is having at both the ends, then we are calling it as amphi. Amphi means both. The flagella at both the ends of the bacteria are called as Amphitrichus bacteria. And the example for this one is Alcaligenes faecalis. Next we have Lophotrichus, having a tuft of flagella or the numerous flagella as a bunch or as a tuft at one end of the bacteria is called as Lophotrichus. Example, Helicobacter pylori, that is the bunch at one end. If the bunch is having it at both the ends, then we are calling it as Cephalotrichus. Lophotrichus examples, Helicobacter pylori, Spirillum and Pseudomonas. As I told, a bunch or tuft of flagella at one end, and if it all present at both the ends, then we are calling it as Cephalotrichus. And the example for the Cephalotrichus is Streptococcus, the bacteria Streptococcus. The last one is Peritrichus. Now in case of Peritrichus, the arrangement of flagella, here the flagella are arranged throughout the body. You can see here throughout the body of the cell, all are directed in different directions or different ways. Now examples for Peritrichus is Bacillus subtilis, Salmonella, Klebsiella. In the Peritrichus arrangement, if you see, the flagella form a bundle that moves the cell towards the stimuli through the run movement of the cell. These flagella rotate in the clockwise direction and hence cause the movement of the bacteria. Whereas in case of Cephalotrichus, which are having the flagella at both the ends, and also in case of Amphitrichus, that is single flagella at both the ends, when we are talking about the movement or the mechanism of movement, they operate differently. That means these flagella, they work simultaneously but rotate in opposite directions. However, the mechanism of the movement is similar to the arrangements. When we are talking about the movement in case of Lophotrichus, the multiple flagella which are present at only one polar end, they have to move in the same direction and with the same speed so that they can move in a smooth movement. And these flagella are controlled by different motors because the motors are triggered by similar stimuli and thus causing or generates the flagella to occur together. In case of monotrichus, the movement is counterclockwise movement of the flagella. This movement pulls the bacteria backwards and allows the reorientations. Because of the counterclockwise movement of flagella, it will allow the bacteria to pull it backward and also allows the reorientation of bacteria. Thank you.